Okay, so I'm Ms. Rainier, and I want to explain to you transcription and translation with my model. So what I have here is my DNA, and I have my promoter region here, right? And you can see my DNA is double-stranded. So the first step that I need is I need the enzyme RNA polymerase. So that's my RNA polymerase. Is going to need to recognize that that's the promoter. So typically there are transcription factors that will bind to the promoter, right? That region just before my transcriptional unit, my gene. And then my RNA will bind there and it will pry open the DNA. And as it does that, normally it would add one base at a time. That's a little bit hard for my model. So what I'm going to do is I'm going to have it add in a couple codons at a time, and it moves down. The next one comes in, matching up C with G, U with A. Okay. RNA polymerase continues to move down. Okay. Next one moves up, and I should say that as it goes through, my RNA that's being made starts to be a single strand, and my DNA starts to go back together. Okay, so it's another little limitation of my lab here, of my model here as well. All right, so my RNA polymerase continues on. All right, my DNA goes back together. Here's my mRNA. Continues on, opens that up. Next one going in, matching up. Make sure I have my, whoop, it's a little bit funky there, but we'll go with it. And then continuing on. All right, opening it up. Right. Again, move my RNA down a little bit. The DNA goes back together. It continues on. It's the last segment in there, my RNA. And my DNA goes back together. Okay, and then my RNA polymerase would just fall off. Okay. All right, just seeing how much time I have. Okay, so here I have my RNA, and notice that I have a section here that's a little different color. That's going to be one of my introns that needs to be cut out. But before that happens, I need a methyl G cap that's added to one end. Right, so to the 5' end, there's my 5' end here. And then to the 3' end, it gets a poly A tail. Okay, normally that poly A tail is going to be much longer than that. Again, just kind of the limitations of my model. Then I'm going to need, okay, so part of my processing that's taking place now, so I have my processing. Besides my methyl G cap and my poly A tail, I also have my spliceosome that consists of multiple SNRPs that act as enzymes, and they recognize what should be cut out from my RNA. So it splices out this intron and it splices back together the exons. And the thing to note here is that there can be multiple introns, or another time when that RNA gets made, it might not cut out any introns. So this one gene can actually code for multiple polypeptides. So that's pretty cool. So that's alternative gene splicing. So let me just get this out of our way. Let me also recognize that there will be an untranslated region. I didn't put that as part of my molecule. Usually that's going to be in my DNA. Before, whoops, the 5' end. Hang on a sec. Okay. I'm just moving my RNA down a little bit to give us a little bit more room to do our next step, which is translation. So my processing is complete, and now this ribosome, this RNA, messenger RNA, needs to exit and leave. I should say I forgot to put the 3' untranslated region in as well. So let me do that. Okay, just to show you just my finished mRNA. All right, so now it goes out to the ribosome. The ribosome consists of two parts. There's a small ribosomal subunit, and there's a large ribosomal subunit, okay? And mine works a little bit differently. Normally they would glom on together. But what happens first is that small ribosomal subunit is going to locate where that first codon is, where that start codon, the AUG. Now, meanwhile, also in the cytoplasm, I have another kind of enzyme called tRNA amino acid synthetase, and that's going to put the amino acid onto the tRNAs. So over here you can see I have my amino acids. They look like little balloons, and then I have some really kind of cheesy looking tRNAs that are just showing the anticodon, all right, just to make it more simple for my model. All right, normally these would consist of, I think, about 89 or so bases, okay, but I'm just showing the 3 that are going to come into play and match up with the codons on my mRNA. So my tRNA amino acid synthetase, what its job is to match up the amino acid with the tRNA to get the correct one on there, so the correct tRNA is going to be brought in. We're going to require some ATP to do that, okay, so some ATP, or as I like to say, some ATPD, all right, a little homage to Dustin Pedroia. All right, so when that gets used, that becomes ADP, all right, that provides the energy to do that, to put them on. And then my tRNA can direct into this spot, right? So I still need my second part of my ribosome, my large ribosomal subunit. That would typically come in and rest in what we call the P site. But to make it a little easier, I'm going to put mine down below. But we can hopefully see here that the ribosome is going to read the mRNA 3 bases at a time, or 1 codon at a time. So this tRNA is sitting in at what we call the P site. The A site is open to receive the next amino acid. So if this is CAC, the anticodon for that is going to be GUG. I would also be able to use my little chart, my handy dandy DNA decoder of science, to be able to figure out that CAC is coding for histidine. Okay, so GUG is going to be the anticodon. I would need my tRNA synthetase to be able to join those together. This would come in. It would match up here. And because the ribosome is made up of RNA, it can also act as a ribozyme. So it can act as an enzyme that's able to catalyze the peptide bond that's going to form between the 2 amino acids. So I like to sort of show that as requiring, whoops, I've got something on top of this here, requiring some GTP to do this. It's going to provide energy for that bond to be able to make, or be made, sorry. So I always kind of think it makes a little ka-chunk sound. So ka-chunk, those bonds are formed. So a peptide bond here, and when that happens, now the methionine, that first amino acid, is not attached to its tRNA anymore, and the ribosome is going to translocate down 1 codon. So now this E site has a tRNA that has no attached amino acid to it, so it's going to leave. All right, and now the next one will be read, the next codon, GUG. If I look up on my chart, GUG is going to code for glycine. So I have my glycine here somewhere, hopefully, yep. So GUG, I need to have CAC. Yep, sorry, not glycine, but valine. So that's going to come in. CAC matches up here. All right, we know we need some GTP, ka-chunk, binds those together. All right, and there's where my magnets get not so magnetic. My ribosome translocates 1 codon. This tRNA leaves. It can go pick up another histidine. We're going to read CCC. That's going to match up with GGG, that anticodon. So that's proline. That comes in, matches up. We know we need our GTP, right? Ka-chunk to make that peptide bond between the proteins, between the amino acids. We have translocation that takes place. This tRNA leaves because it's in the E site. It exits, right? And we're going to keep going down. The next one, UUU, is going to be AAA. That should be phenylalanine. That's already been put together. We have our GTP, ka-chunk. So lots of energy being needed to do this process, right? Peptide bond forming between them. This tRNA exits. Then I'm going to read CUC, so GAG, I'm looking for that for my anticodon. It's going to come in. Of course, it would have been put together by the tRNA aminoacyl synthetase. Need some more GTP, ka-chunk. That's going to form that peptide bond. Right, so now it's attached here. Moves down, translocates another one. This tRNA exits. Notice my growing polypeptide chain, right? If it goes down, I just did CUC, so I have AAG. So UUC, that's lysine, right? That would have been put together. Back at the, in the cytoplasm at the tRNA aminoacyl synthetase, that comes in. It's held in, held in position, right? The one that has the growing chain is at the P site. The next one comes into the A site. GTP, ka-chunk, right? Now that's bonded to the leucine is bonded to the lysine. Translocation, this amino acid exits. This tRNA exits. And then I'm going to read GGG. Anticodon is going to be CCC. Happen to have my glycine, comes in, it's going to bond. We're going to need GTP, ka-chunk, right? Ribosome moves down. Now we've entered into the A site, UGA, right? That's a stop codon. So instead of coding for or calling for a tRNA with an amino acid, instead what happens, a release factor is put at that site, and a water molecule is used to be able to allow the bond between the amino acid and the tRNA to be complete, and it allows the whole complex basically to be released. So the RNA subunits come apart, small ribosomal subunit, right? And now I have my polypeptide chain, and if we think about where that will go next, that will head out to the Golgi body to be maybe cleaved up, folded up, maybe some other chemical tags added to it. Okay, so that's transcription and translation, and I hope that made sense.